Okay.
Hola, muy buenos días. Nos da mucho gusto darles la bienvenida a esta tercera serie de los seminarios en Sistemática, Evolución y Biodiversidad del Instituto de Biología. Esta es, por tercer año, tenemos esta serie donde invitamos a ponentes seleccionados por nuestra comunidad para que nos hablen de algún tema en el que estos invitados son líderes científicos a nivel internacional. En esta ocasión vamos a tener 8 ponencias con diferentes temas, iniciando el día de hoy y terminando aproximadamente el día 23 de mayo. Estas conferencias se llevan a cabo los martes a las 11 de la mañana en el horario normal de nuestro seminario del Instituto de Biología y nos da mucho gusto y los invitamos y las invitamos a que participen en estos seminarios, a que escuchen, a que aprendamos todos, que hagamos preguntas y que interactuemos con los y las ponentes. Sin más que decir, reitero la bienvenida y le doy la palabra al doctor Marcelo Pase, quien está organizando esta transmisión como parte del comité organizador. Adelante, Marcelo.
Sí. Muchas gracias, Susana. Sí, en el día de hoy, entonces, como la invitación a Tom Gilbert partió de Alejandro Ceguera, va a ser quien va a presentar nuestro ponente. Y ustedes pueden ir haciendo sus preguntas en el chat y después al final las vamos a transmitir. Alejandro.
Yeah, thank you very much, Susana and Marcelo. Good morning, and thank you for being here. Today we're pleased to welcome Professor Thomas Gilbert. Dr. Gilbert is the director of the Center for Evolutionary Hologenomics of the Danish National Research Foundation, as well as the group leader of the Hologenomics group at the Globe Institute of the University of Copenhagen in Denmark. Professor Gilbert obtained his PhD at the Department of Zoology at University of Oxford in 2000. Then he did a first postdoc at the University of Tartu in Estonia, and then a second postdoc at the University of Arizona. Since 2005, he has been associated with the University of Copenhagen. And at the beginning, he was deeply involved with the study of ancient DNA, and without doubt, he's a leader in that field. And besides that, Dr. Gilbert has been involved with very relevant studies on human genomics and evolution, or monitoring the biodiversity through the sampling of environmental DNA, and so on. Recently, he has been developing and promoting the idea of hologenomics, and that is exactly the topic of today's talk. So thank you very much, Tom, for accepting our invitation. It is a great pleasure to having you here in the first 2023 seminar of Fronteras en Sistemática, Biodiversidad y Evolución. Thank you. Please.
Thank you. I will try and share my screen. I hope we're good.
Yes, it's working well. So
excellent.
Thank you.
Well, firstly, thank you so much for inviting me. It's a real privilege. I've had many students and postdocs from UNAM in the past 15 years. One of my first PhD students was Maria Avila, and she set the way for some really incredible people that we had the privilege to host here. So it's always super to be able to do anything to say thank you to UNAM. So I'm delighted to be here, and thank you to the organizers for asking me. And I'm going to talk about evolutionary hologenomics, which is the area the center I'm in works on, and hopefully to give you a feel for why I personally find it so exciting. But for the record, as briefly mentioned, I am an organismal biologist. I was convinced when I was an undergraduate I was going to study animal behavior. For example, actually this fish here, Rivulus, how this fish is able to navigate out of water when the streams dry up. I was very into that kind of stuff, very much not interested in genetics or microbiology. But in my PhD, I kind of learned differently, and I worked on various ancient and modern DNA projects on organisms, big organisms. I was very much still avoiding the microorganisms. And then, of course, in about 2006, when the first next-gen machines started to come out, like the GS Flex, I was lucky enough to get my hands on one of the first 454 machines and started to try and move into the world of genomics. And actually, this is why I was so happy to meet Maria Avila, because I knew nothing about computing and programming. And Maria had just been trained in the genomic science program and basically came and saved me. It's fairly safe to say, had it not been for Maria, I would have never managed to make my way in the world of genomics. But of course, those who can remember this far back will remember that back in 2005, it was very expensive to do genomics. The outputs of the machine were very low, like a GS Flex or a 454 would maybe get 20, 40, 50 meg of sequence in a run for a crazy amount of money. And of course, it was very much therefore the domain of the very, very rich, which was essentially medical genetics, crop genetics, applied animal genetics, and so on. But luckily, I got to experience the technological revolution, with the key output being that the costs of genomics dropped, and eventually people who are essentially interested in ecology and evolution of not things that are very money, they don't raise a lot of money, could start doing genomics. And that's what I basically started doing, classic ecology and evolution with genomics. And just to give you an example of the kind of questions I was working on at the time, there would be things like this. How does a vulture, how is it able to eat the rotting carcass of whatever it finds on the road? How is it adapted to do that? And in my mind, the answer had to lie in the genome of the vulture, some kind of gene and super immune system to allow it to do this. And in fact, a Korean team even published this nice paper studying the genome and the transcriptome on the vulture to show how this explains how it manages to fill this niche. Other questions I've been very interested in are things like this. Why, if there are 10,000 or so birds, are about 65% of them the passerines? What is it about the passerine lineage that made it so successful? And if you'd asked me 10 years ago, I would have said something in their genome or some things in their genome give them the ability to diversify and radiate. Another example, I worked for many years with killer whales, thanks to a guy called Andy Foote who joined me. And one question he was very interested in is how are killer whales sympatrically speciating off the coast of California? You can see in this figure here, there's a phylogeny on the right with types B1, B2, and C. And Andy was working on them and seeing that they lived in a very similar area. They did differ on the diet, but they appear to have no gene flow between them. And so what was causing these barriers? And actually at the time we sequenced their genomes, and we actually argued for something we call genome culture coevolution, essentially the formation of culture in it causing a reproductive barrier. And another question that I still am very interested in, how did we domesticate what is essentially a dangerous predator specialized in hunting large things into the dog, right? Which is to be very blunt, you would not put your baby next to a wolf. You will quite happily put your baby next to most dogs, right? How did we change that? And of course, there have been many genomic studies where people have compared the genomes of dogs to wolves, and they've identified many genetic differences, and they've argued these have got relevance to the process. So classic genomics. Oh, sorry, and a last example actually relevant to Mexico that I have been thinking about. What is it that drives lineage distribution? So here's a very simple figure showing the distribution of various maize land races, and they're quite different. Why is that? Well, the classic example, of course, would be genetic adaptations in the different lineages allow them to grow in the different conditions. For example, the drier conditions as you go north are probably linked to certain genes in the genome. And just to be 100% clear on this, I still very much believe genomes are super important. I'm a very big believer in the Earth Biogenome Project and the related initiatives that are arguing that the world needs very high quality, standardized whole genome reference sequences, sequenced in the same way, annotated in the same way, publicly available, because that is, I think, going to have an enormous benefit on our planet in many ways. And I fully support this and will happily talk about it anytime. But as has been mentioned, my interests started to change in the last few years, and now I do run a center that studies the holobiont and the hologenome. And I'm sufficiently excited about this. I will take any opportunity to talk about it and try and argue that this really is the way we need to study life if we go forward. So what I propose to do is talk about what I think a holobiont is and what its hologenome is, for those that are maybe not familiar with the term. One way of describing it, to paraphrase Dr. Spock from Star Trek, is it's life, but not quite as we know it, because it really is quite logical when you spend a little bit of time thinking about it. And actually, it's something that we actually have known about for a long time in certain areas, the microbes and the roles they play in certain systems. But what I hope is to also present a few contexts that are maybe more surprising. And so after having done that, I will present my ideas, at least, for why I think it is relevant in reinterpreting ecology and evolution. And just for the record, I am very happy to be wrong. I am fortunate enough to have got to a career stage where I can afford to consider what other people think might be outrageous ideas. And I'm always happy to be shown I'm wrong, but I'm also happy if people want to go out there and prove I'm right, especially if I don't have to do the work. So my journey into the holobiont started back in 2013. For nearly 12 years now, I've been running a very large bird genomic consortium called the B10K project. One of our key collaborators is this man, Gary Graves from the Smithsonian, and Gary likes vultures. And back in about 2013, he wrote to me and said, Tom, I have this amazing opportunity to catch a lot of vultures in Tennessee and actually sample them to see what microbes are on the face and are inside them. And do you want to do some work on that? And I was like, why would I possibly want to work on that? I'm a genomics guy. I don't do microbes. And Gary said to me, well, have you ever thought about how a vulture eats a carcass? And I hadn't. And I said, no, Gary, I haven't. And Gary told me what I found to be quite remarkable, that the beak and the claws of the vulture are not sufficient to force their way inside a carcass of a deer, for example. What they have to do, they have to enter through the soft parts. They have to stick their head in the mouth, in the eyes, or up the anus of the rotting carcass. And Gary was like, think about the microbes it encounters in this situation. Shouldn't we look at what microbes are on the vulture? Now I'm getting them. And I was like, I guess so. Sounds kind of interesting. And I hadn't done any microbiology. And I was aware that this genome paper had come out explaining how the vulture lives on the carcass. So thanks to a collaboration with a microbiologist colleague of mine that I knew, we did what was my first microbiome study, 16S sequencing of the facial skin and the hindgut content of 2 vulture species. And we produced what everyone else produced, essentially a pie chart showing some distributions of certain things. But what was striking was, if you can see, there is like a large purple of a fusobacteria and a fairly large green of Clostridia. These are pretty nasty pathogens that are associated with very much rotting carcasses. And you could say that's not really very surprising, because what is the vulture eating? It's rotting carcasses, and it's putting its head inside a rotting carcass. But we did do one thing that I think was very clever in retrospect. We had a control vulture from Copenhagen Zoo that has only ever eaten fresh chicken. It derives from many generations of vultures that only eat fresh chicken. And yet its face and its gut is full of the same Clostridia and fusobacteria. And this is weird, because basically it's been maintaining these very nasty microbes on its face for generations. So it's not just getting it from the food. And Lysandra, who was one of my many talented Mexican PhD students, actually then did a proper shotgun metagenome study and looked at what microbes were in there and came back and said, look, I mean, these microbes appear to have the potential to do some very interesting things, like forming resistance barriers to colonization, or there are predatory eukaryotes in there, or there are phages, and so on. And she was basically saying, look, I mean, I basically think these are being maintained because they're actually part of how it fills the niche, right? What basically we were seeing, and this was very revolutionary for me, simply because I was not a microbiologist, we were seeing what was a holobiotic basis for adaptation to rotting carcass. Essentially, sure, the vulture's genome does something, but also the genes of the microbiome are critical to filling that niche. And only if you have both together, the holobiont, the union of two, can you actually answer this question of how is the vulture filling the niche? And so this is the terms. The holobiont is this term, which is quite an old term. It was proposed in the 40s, but it's really in the past 20 plus years that it's been used more commonly, that we have to view organisms, especially larger organisms, as a union of the host and the microbe. The host is maybe the scaffold, the microbe is a key player. And the hologenome is the combined genome of both. And basically what we believe in is if we want to answer any of these key questions in ecology or evolution or even in medicine, if we don't study the hologenome, we're never going to see the full picture. So now some of this is really obvious. I mean, there is a lot of interest today in the role of the microbiome in the applied sciences. I'm sure many of you have encountered news articles or scientific articles about all the many, many ways microbes affect humans. Yeah, pretty much every disease these days is being linked to it. It affects behavior, and it affects like obesity and arthritis and so on. And there is a very large probiotic industry right now trying to improve the health and the growth and the efficiency of animal and plant production using microbes, right? So that they have certain health-related roles and food digestion roles, of course, is not new, right? Not new at all. But what is more interesting is when you start to look at the diversity of studies that are coming out on all the other things that microbes are being linked to in animals, right? That are not simply a cow digesting grass. I mean, here are just some examples from a paper we wrote a while back, but foxes, for example, are being argued that the reason foxes can urbanize in Europe and essentially adapt to a very plant-rich diet, essentially eating pizza crust and that kind of stuff, is thanks to their microbiome changing. Their microbiome is allowing them to open this into this new niche. There are Neotoma wood rats, which have changed what they eat, and there were plants that were toxic to them many generations ago are now completely edible by them because they've modified their microbiome to deal with the toxicity. There is a remarkable study that basically sampled the fecal microbiome by basically taking it from inside the intestine of active and hibernating brown bears. They find a difference in the microbiome, and then they actually transplant some of the microbes into mice, and the mice basically start to exhibit signs of hibernation. So the hibernation is being linked to the microbiome. There are examples about temperature preference and behavior and so on. And these are all pretty strikingly beyond, let's just digest certain food. And this is super interesting because what it suggests is that the microbiome is a mechanism for increasing the flexibility of the organism. So if you think about what we're all told about how does an organism adapt, well, the classic genome-centric view is within a lifetime, you might be able to modify gene expression, some epigenetic tweaks, but basically to really adapt, you have to have babies and, get genes from the father and the mother and create new gene mixtures and select. And it's a very slow process to adapt through genomes. But the beauty of the metagenome, of course, is you can change your microbiome almost instantaneously. And if microbes are bringing in this crazy range of possible parameters into your life, it's a very, very, very fast mechanism for adaptation. And so in light of this, if you think about a classic Darwin-like quote, it's not the strongest of species that survives, nor the most intelligent, but the ones most responsive to change. The relevance of this is obvious. For the record, and I just like to say this, Darwin never, ever, ever said this quote. Many of you might know this. Darwin was 0 years old in 1809. That's one thing. This quote comes from a guy called Leon Megginson, who was a business studies professor who liked Darwin very much, but for some reason, people have really latched onto this. What Darwin wrote is a lot less well written. I'm not going to read it out here, but the sentiment is the same, right? It's about what can adapt fastest. So in light of this, actually, my journey into hologenomics basically started with this opinion article we published back whenever in 2016, basically saying, are we maybe overlooking the fact that the gut microbiome of vertebrates might be a super important mechanism for rapidly adapting, and maybe one we should be taking seriously? Now, I actually take it so seriously that if I return to some of the questions I was interested in genomically, I actually believe I can propose alternate explanations for all these things about what was going on to what I used to propose that are based around the holobiont. So I'll give you some examples. If I go back to my sympatric speciation in killer whales, as I mentioned, the different ecotypes have got different diets. One is a bird eater, one eats marine mammals, and the other one, I think, eats fish. Why is this relevant? Well, one thing, of course, we know very well now is that as you change your diet, you have a very big effect on the gut microbiome. This is data on humans, sadly, not killer whales, but the premise is very well known in different animals. This is relevant if you think about the interest right now in the gut-brain axis, essentially the idea that microbes in your gut are taking what you eat, pumping out chemicals. These affect the brain, and these can have consequences. Because I would propose an alternate hypothesis for this sympatric speciation is that they have different diets that give them different microbes that have differential effects on the brain that basically lead to this reproductive isolation, or what we called culture, right? It's actually driving a preference to basically hang out and reproduce with the guys with the same kind of production. I have no proof of this, but I think it is a way of explaining this like weird cultural isolation where the killer whales are clearly not interacting with each other despite living sympatrically and encountering each other in the same geographic area regularly. Is there any evidence for this at all? I was very excited to find this paper called Diet, Gut Microbes, and Hostmate Choice, which actually did suggest maybe there is a link. I was slightly less excited to find it was on Drosophila, but it still is the same kind of principle, right? They're actually seeing that the Drosophila are picking mates based upon the gut microbes. And actually, the more we look at behavior, we see funny things. Here's just another example. This is a work that was done by Mads Bjørnsen, who was a master student. He went to Greenland, where we have sled dog packs, and he went to 8 packs, and every pack has what's called a lead dog. It's like the one the owner thinks is, it's kind of like the owner's favorite, and it sets the way and the others follow it. And it's picked based upon the owner's feeling for what is the best dog. It's not like a set parameter you can measure. And he also sampled the microbiomes of the followers, and he sampled in particular the oral microbiome and the fecal microbiome. And although the 8 packs were fed very different things, and although the owners basically said they gave no preference to the lead dog with regards to food or so on, and although the oral microbiome showed no pattern, when you look at the fecal microbiome, while the diversity of the microbes in the fecal microbiome of the followers ranges from quite low to quite high, you can see it in green here, the lead dogs always have a relatively low diversity, regardless of which pack it is, what location it comes from. And this is kind of interesting. And actually, when you then ask the owners, do you have any follower dogs which you think have got leadership potential? They're always follower dogs with low levels of microbial diversity. So We can't explain what's going on here, but there is, a pretty strange, striking pattern. Here's another data set. Lara, who is a PhD student, worked on a cohort of chickens that were generated in a selection experiment where red junglefowl were taken for 6 to 10 generations and selected on to be more tame-like and more fearful of human-like. And the man doing this in Sweden has been doing genetics and epigenetics to look at what's going on in the genome as they segregate. Lara sequenced the gut microbiomes of these and basically came up with a figure of this showing there are significant differences in the relative abundance of certain microbes, despite the fact they're fed the same diet and they're housed in the same conditions. And actually, when you look at, for example, the effect on gut-brain modules, which is just genes in the microbes that might be functionally important, she basically sees there are differences. For example, in the more fearful ones, there is kind of certain genes linked to a potential production of anxiety and fear and so on, whereas in the blue ones, which are the chilled out, relaxed chickens, there are certain microbial genes linked to fear reduction, fear extinction. So they do appear to be correlating. Their gut microbiome, despite getting the same food, is separating with the behavior, raising a question of what's going on. And if we go back to dogs and wolves, so as I mentioned, lots of genomic work has been done comparing the wolf genome to the dog genome and identifying many genetic differences which explain why a dog is not a wolf. But what most or all of these studies ignore is the question of how did we get through the first part of the process? We know how the end product is different, and we know why dogs are dogs today. But of course, people didn't take in wolves and on generation 1 had a fully formed dog, right? It took a long time to select on the genetic variants in the wolf to end up with the dog. So how did our ancestors coexist with the wolf when it was basically still a wolf, which is something you absolutely cannot do today, right? Well, I argue you need some kind of buffer to keep the wolf more amenable during the selection process. And I actually wonder if the buffer, again, is linked to the gut-brain axis. And basically, maybe when our ancestors first got hold of wolf puppies, maybe, what do puppies do? They lick their owners and they eat any human feces they can find and so on. And maybe that basically modified the microbiome of them, modified the brain to make them a more relaxed, chilled out animal, which we could coexist with for the generations needed until we'd finally finish selecting on the genome for the permanent changes. Sounds crazy, but if you're a dog owner, you will know dogs love to eat human feces. It's disturbing how much they like it. They preferentially will take it over dog feces. But again, there is also some interesting data. So Lara, who I mentioned before, she also has worked on the Belyaev foxes. These are the foxes that Belyaev in the Soviet Union selected on over multiple generations for a more tame-like, dog-like form and a more fearful, aggressive form. They've been studied in great detail. What Lara did was get hold of their fecal microbiomes. Now, again, the key thing here is they're raised in the same facilities. They're fed the same food. They are allegedly not handled. Allegedly, kind of hard to know what happened over 50 years. And Lara sequenced the gut microbiomes of them. And basically, again, this shows gut-brain modules. And basically, the top yellow ones are the fearful form and the blue ones on the bottom are the tame form. And the things in blue are basically significantly different gut-brain modules. And when you actually dig into what they are, well, they're linked to things like glutamate production, which is well known to be linked to changing aggression, and butyrate, which is well known to be linked to calming in other species. So again, like with the chickens, we're seeing this parallelism of behavior change with the gut microbiome. Now, a classic question is chicken or egg, which came first? But one thing I did find that I really find interesting, this paper, which was by Anna Kukakova and Gorgi Zhang, that was the genome analysis of the foxes of the different lines. They talk about many genetic differences they find, and they're all obvious, like brain-related stuff and so on. But I'm highlighting in red here a half sentence that literally this is all the thing that appears in the paper, and it's like, wow, we also see some differences in the immune system, and they stop at that. What I'm wondering is is essentially some of the initial selection affecting the immune system in a way that's affecting the microbiome, and then bingo, the whole situation takes off. So I have, this is the kind of interesting thoughts you can have. So, and another question just linked to this that I've often thought about: why is it our ancestors domesticated the wolf, but none of the other crown canids? I mean, humans probably domesticated the wolf because it was probably useful to have an animal to help hunt and help guard, but that happened in Eurasia maybe 20,000 years ago. What about the hundreds of thousands of years in Africa when you need to hunt and you got lions and stuff that will come at night, and you've got all these other canids which hunt and have got social-like behavior and so on? Wouldn't it have been nice to domesticate them? Now, of course, on the one hand, you could give a kind of technological argument that, well, humans hadn't really thought about it and weren't clever enough. But another possibility is, if my hypothesis is correct, that it required some kind of microbiome transplant, could it simply be that the microbes needed to affect the wolf are simply not capable of coexisting, living inside the holobiont of the other canids? Maybe they could eat all the human poo they want and just simply not pick up the microbes. So this actually is an idea we kind of like to conceptualize under the idea of viewing the microbiome. And here I'm talking about gut microbiome, but to be honest, it can be any associated microbiome, as a phenotype of the genome, just as how height is a phenotype, or eye color, or what else. And the way I have this idea is, imagine in any environment, you knew every possible microbe. And how you define microbe is complicated. We can get back to that later. But for simplicity, just imagine, every single microbe that could exist in your environment. And could is not the same as does. They may be present or absent, but just imagine you could lay them out in front of you in a matrix. And let's imagine in this example here, white is it's not present right now, and black is it's very present, and gray is a frequency of abundance. Well, I like to think of an idea I call microbiome space, which basically, in the context of an organism, would be which microbes could live and interact with an organism in an environment and which ones can't, simply because of the way the organism is. So, for example, a cable bacteria, which do not live inside animals, would not be inside the human microbiome space. But Fusobacteria, which is pretty nasty but can be found in it, could be in there. It might not be in there, but it could potentially be in there. It's a way of delineating which range of microbes could be in there. It's very similar to, I guess, in ecology, you can think of the species that could be in an environment versus actually are in an environment. Now, obviously, the environment has a massive effect on which microbes are there at any time. So at time equals 0 inside host A, you might have this mixture. At time equals 1, it might have changed. You might have, drunk some coffee or something, and time equals 2, it might have changed. But the point is, it's still a constraint of the host genome which microbes are there to start with. And what we're very interested in is, how does genetic variation between individuals shape this space and shape the overlap versus the unique parts of the space? Because I think this has consequences for adaptive potential. If, for example, as you can see on the right here, there is a certain microbe that you need if you want to enter hibernation, and it's not compatible with host genotype A, they haven't got that chance of ever using the microbe to go into hibernation, and so on. I would like to note that genome constrains the microbiome is pretty obvious and actually explains all sorts of fun observations. This is data from a study looking at the microbiome of the gut of the panda and comparing it to various other bears and carnivores and herbivores. And the key point is, although a panda spends all its time eating plants, it has failed to evolve or adapt to the microbiome needed to eat plants. It has the microbiome of a carnivore, basically, which is why the poor thing has to eat constantly, because it's just not getting any good out of the bamboo, right? And it's been trying for a very long time, but its genome is constraining its microbiome. It's stopping it getting to where it wants to be. So you can ask the question, Tom, does genetics shape the microbiome at all, or is this crazy? Well, obviously, interspecifically, there's no doubt. If you just compare the gut structure of different animals, they're very, very different. And gut structure will have an obvious effect on the microbiome. So nobody will disagree that the genetics of a rabbit or a human is not relevant to their gut microbiome. If you care about plants, it's the same thing. Different root structures will have a very, very different microbiome. Why do they have different root structures? Genetics. Intraspecifically is much harder to say, partly because people were very obsessed with humans to start with. So this study, for example, studied the gut microbiome variation of different humans, from hunter-gatherers in Tanzania to pastoralists in East Africa, and West Africa, to Europe and the USA. And the gray scaffold just is the same thing, and the colors just show the different abundance of bacteria. And of course they're different, because the people here are eating very, very different things and living in different places. Humans are a shitty model to use. Here's another example from plants. This one shows the different microbiome of the roots of different barley land races in Israel. Sure, they're very, very different, but of course they're growing in very, very different environments. So it doesn't teach you very much. The human guys did try some clever studies on twins about 10 years ago, looking at the microbiome of identical versus non-identical twins. I got told off incidentally. Apparently the Olsen twins are non-identical, even though they look identical, but pretend they're identical for now. The logic is, if twins are raised together, they're in the same environment, they get the same food, and they do find that the identical twins whose genomes are identical have a more similar microbiome than the non-identical twins. But it's, humans, very, 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 very tricky. But actually, completely independently, we actually kind of stumbled across a bit of evidence on this. We were working on sand eels, which are just a very heavily fished species in the Baltic, doing regular population genetics on it. And we see that outside Copenhagen, where the green line is, there's very different structure in the sand eel population because the salinity of the water changes. But Kathy, who was doing the work, also actually sequenced the microbiomes of them, and she found this big difference in the microbiomes correlating with the genetic structure. Now, if you're clever, you'll be saying, well, of course they were. The water was completely different. It was high salinity on one side and low on the other. But when you overlay the water microbiome on this, you don't see that. So you do start seeing these very clear structuring effects alongside genetics. And this is actually much easier to do with plants. One study a master student ran, Mads, he grew different maize land races on exactly the same soil in Denmark. And he, after 3 months, studied the microbiome on the roots. And basically, you find things like this. This is just a measure of diversity between, in this case, Ancho, which is a Mexican land race, and B73, which is a very, very dominant maize land race. And after 3 months on the same soil, they show very different characteristics. So they've been selecting for very, very different microbes from the standard stock of microbes. And when you put in more land races, you see the same kind of effect or so on. And this then raises me to speculate, does this genome-based selection on the microbes in the soil actually maybe explain the distribution of the different land races, as opposed to a classic answer, which could be some kind of gene to do with light or flowering time or so on? Now, in this modern era, there's a nice way you can start testing this. You can use CRISPR. So Morten here is a colleague of mine. He basically looks for people doing CRISPR on animals for any reason, and he asks if he can then sequence the gut microbiome, and the results are pretty astounding. So this is one example. There's a group making albino zebrafish. They're just knocking out the skin pigmentation. It's a single mutation. They sequence the gut microbiome of these two morphs that are raised together. They make a PCA, and look, they get completely different gut microbiome, even though it's a mutation affecting the skin pigmentation. When you dig into it, it turns out that the skin pigmentation actually starts with the transfer of some molecule in the gut and so on. But the point is, this single mutation that has nothing to do with the immune system is significantly affecting things. And the fun thing now, of course, is you can do this in all sorts of cases and really start to dig into the effect of what's going on. So going back to my canid question about why is it the wolf maybe was able to take up these behavior modifying microbes, and why not the other canids? If you buy into my microbial space idea, maybe we have a generic canid microbiome space. Maybe the wolf one is slightly different, and maybe the microbes needed to change the behavior fall in the wolf space that is unique to the wolf, which means that no matter what other canids try and eat the feces and get the microbes, it just doesn't establish and doesn't have the effect. Whether that is the case is another question, but at least this idea of microbiome space, you can start to come up with hypotheses. If I go back to my early example of why are there 6,500 passerines and, not of all the other birds, maybe the generic bird microbiome space is this, and maybe the passerine microbiome space is much, much bigger. And maybe that just allows it to utilize a much wider range of microbes to diversify and eat all sorts of other interesting things. Is there evidence for this? Not direct, but I did find this one paper that is by no means perfect, but it just shows the diversity of different and the relatedness of different avian order microbiomes, and passerines are in yellow and they're all over the place, right? Now, of course, they have very, very different diets, which has a confounding effect. But this is also the kind of thing I would expect to see if I was correct. And going back to maize, I mean, we have these different land races. Maybe if you really get down to studying the microbiome space, you end up with something like this. The yellow one, which is fairly unique, falls into an independent area, and the other ones overlap to some degree or not. And B73, which is notably super successful at growing everywhere, maybe it has the absolute massive microbial space. Maybe what's important is selection on genes that allow it to work with a very diverse range of microbes in the soil, therefore grow in a very diverse range of places, which is quite different to other explanations for what the genetic basis is of what's going on. So what is the point? Well, my first point is microbes do confer very surprising properties on their hosts, and there's more and more stuff coming out all the time. I really do believe they can be shaped by host genomic variation in many, many ways. And the differences in which microbes hosts can maintain could have very surprising consequences if you think about it. And going back to my early attempts to study evolution with genomes, I mean, what I was really falling for is the classic partial insights and missing the whole, right? We were very convinced we had answers, but ignoring the microbes probably means we haven't really got even slightly close to what was going on. So I'm going to end now with an idea I'm playing with in my mind. This is purely a thought experiment, but it relates to an interest I've had for a long time, which is what happens during extinction. And so I call it extinction genomics versus extinction hologenomics. This is not a published idea. It's literally a thought experiment, but hear me out. I had this grant from the European Research Council called Extinction Genomics, which was very much linked to my interest in ancient DNA. And basically, we were studying what happens to species when they either go through big bottlenecks or they go extinct. What happens to their genomes? Were there genomic predispositions? So basically, we would sequence genomes of time series of samples. And if I dig into the answers, were there genomic predispositions for which species go extinct or not? Not really. Not that surprising, but not really. What happens to their genomes when they go through bottlenecks? Well, surprise, surprise, they lose diversity. And we could see very nicely how they lose diversity, but not a huge surprise. And why was the diversity important? To be honest, I am no closer to that answer. Why is it important? Or I wasn't. Now, however, I think I have a hypothesis. And this is my extinction hologenomics idea. Let's assume that the genome does, in fact, shape the microbiome space of a species. There are different microbes in there. And let us assume that this space controls the potential for microbe-linked adaptive functions that might be beneficial, whether it is becoming more aggressive or eating a new diet or entering hibernation, for example. Now, let us assume that as our species goes through a bottleneck or the population contracts or whatever, we start to lose genetic diversity in the genome. What if a consequence of that is that it shrinks the microbial space because you are losing genetic variants that would be needed to allow certain microbes to live and interact inside you? Basically, you're now throwing away the ability to actually capitalize on a function of a microbe because you can no longer tolerate that microbe. And as your genetic diversity decreases and your microbiome space shrinks, you're just losing adaptive mechanisms that you could have otherwise used driven by microbes. Is this realistic, or is this just Tom speaking about crazy stuff? I was actually extremely excited to see this paper. This is actually work by a man called Torsten Christensen, who's also in Denmark, who works on Drosophila. And they had found some very nice samples of Drosophila in their freezers that were used in a classic bottleneck experiment in the past. They put the Drosophila through bottlenecks of different sizes, and they measured the effects on the genetic diversity, and they showed that, strong bottlenecks make a lot of loss and so on. But what they went back to do was they went back to sequence the microbiomes of the descendant populations, and basically there is a complete correlation between genetic diversity of the Drosophila and the microbiome diversity, which essentially is the prerequisite for what I'm saying. It does happen to be in Drosophila, as I said, not vertebrates, but it's essentially what I would predict what happens. Can this be tested? I think it can be. There are different ways of doing it. There are many, many challenges, but it certainly is a thing I'm very interested in now, because if you're interested in conservation, there has been this obsession for a long time now about the importance of genetic diversity, and we must maintain genetic diversity. What is less thought about is which diversity. Do we just blindly maximize the diversity in a species, or are there some genetic variants that might be particularly beneficial for long-term adaptive potential? If I'm correct, I would suggest that actually genetic variants in the host that have a long-term effect on the range of microbes you can form your holobiont with could actually be a very important characteristic. So with that, I will say thank you to Susanna, who originally reached out, and of course Alejandro and so on, for inviting me. As I mentioned, I am in huge debt to a very large number of former Mexican PhD students and postdocs, almost all from UNAM, from the biology and from the genomic science program, and of course the guys at Hologenomics who work on this stuff and occasionally agree to test my ideas and spend more time telling me while I'm wrong. And I will stop there. So thank you. Can't hear you.
That always happens. And thank you very much for this amazing talk. It was really, really great. So we have some questions here from the audience. I'll project them on the screen. So the first one from Dr. Yvonne Garçon. Maybe killer whales are just recently sympatric and the cultural differences were obtained in allopatry. What do we know about killer whales biogeography?
Yeah, I mean, Andy Foot is the guy to talk to, but they are definitely very, very recently sympatric. We can see that from the genome resequencing, right? And that's also even more mysterious. They're extremely recent and they show absolutely no gene flow between them. And actually the behavioral observations, apparently, they've been monitoring these for a long time, and they do see them actively avoiding each other. As the pods kind of come into like proximity, they literally just spurn each other and go the other way, right? Globally, there is a lot of structure in killer whales, and most of the other ecotypes show a lot more genetic divergence, right? But they're also, the other ones are geographically, barriers between them, or some kind of barriers. So this is why these are such a weird case, because it's very, very recent. There's absolutely no gene flow going on. So something's going on. We have to explain why they don't have the gene flow. This is where the microbiome could just provide a hypothesis for why they don't like each other, or why they prefer to mate with the different types.
Very nice. The next question, also from Dr. Garçon. Maybe seasonality in the Northern Hemisphere explains why in Africa humans didn't need the fur from dead canines.
Yeah, it's certainly one explanation. Another explanation, actually, that's completely different is that maybe we never domesticated the wolf, but the wolf domesticated itself, because at the time that we think dogs were domesticated, the prey that the wolves, especially the hypercarnivores, were feeding on was getting less and less and less, and maybe the wolves, to survive, started to move in with humans, right? And then some kind of mutualism happened and so on. So yeah, it's quite possible. We don't know. I'm just proposing an alternate idea. In the ideal world, we would of course take puppies of all the different canids and feed them microbes, but ethics, this is tricky. Tricky.
Okay, now a question from Dr. Francisco Vergara. Given the importance you have attributed to holobionts in evolutionary organismal biology, how do you conceive of the relationship between evolutionary hologenomics and eco-evo-devo?
Yeah, that is a great question, and it's not one I've thought about very much. But one of the nice but frustrating things about the microbiome is it's a very intricate system, and we know that microbes affect the host, and we know the host affects the microbes, right? And so it is almost certain that certain microbes inside you, if you're pregnant, will have an effect on the development of the baby and so on, right? And if that happens at that scale, at the greater scale, it probably has a massive effect. But to be honest, this is not a thing I've thought about yet, because we're still stuck on the basic stuff.
That's fascinating. The next question from Dr. Patricia Ornelas. What is known about the relationship of the microbiome? Is there a clear agonistic relationship? I'm interested particularly for the differences in chickens and foxes comparisons.
The relationship of the microbiome in what context? I'm not quite getting this question. You mean with the host? Yeah. Not sure. 100% sure I get that question.
Okay, so I'll move to the next one.
Yeah, but she's very welcome to email me later, and I'll take it.
Oh, Patricia, if you want, you can further develop your question. Let me see here from Dr. Ulises Rosas. Waddington's genetic assimilation proposed that organisms could incorporate new genetic information towards the resulting phenotype. Would you say microbiomes are part of genetic assimilation process? I'm sorry.
There are definitely people that would argue yes, because there are these observations where host genomes have picked up microbial genes, right? There's this example that at least people talk about of like ethnic Japanese carry certain genes from microbes that have got some kind of beneficial effect of digesting seaweed. But yes, I mean, I would absolutely definitely say that in the holobiont context they are, because if you, for example, are a bat, vampire bats, which live in a very weird niche, right? The obligate blood-feeding two species of vampire bat, in order to get into that niche, they had to change their genome, but they also had to change their microbiome, right? And so they obviously had to change in a way with their genome to allow a certain community of microbes to basically exist inside them at the exclusion of the other microbes. So you get this kind of concerted coevolution, right? Now, the fact that the genes in the microbes are not inside the bats kind of irrelevant. The point is they are completely stuck together in this mutualistic relationship, and they can't exist without each other. So I would basically say yes.
Fantastic. So coming back to the previous question, so what Patricia meant is among different groups of the microbiota.
Right. Yeah, sorry. That's a great question. And that's a very complicated question, because how do we actually measure what is positive and what is negative? But simplistically, when people were studying microbiomes, they would just do amplicon sequencing and say, here are the families of microbes that are present, and that's all they would look at. Now the state of the art is to look for co-occurrences, right? So which microbes are appearing together with other microbes, and actually which genes are appearing together is more important than the name of the species, right? And there are definitely patterns of certain groups co-occurring, which would suggest there is a mutual benefit to each other, and also the exclusion of other ones, right? And so if you go back to my microbiome space thing, one of the challenges of that is like, what is the unit in the microbiome space? Is it certain microbes, or is it actually better to view it as communities or agglomerations, or the unit is a group of microbes that work together and form together, and they're a stable construct. And another space would be a kind of perturbation of this, whereas a different construct. And so how you're moving between communities. There is, I think, there is a guy, actually, I read his work. He works on something called the collaborome, which is very much about this, like working out exactly which microbes are collaborating and how they're doing it and how that affects things.
Incredible. Okay, our next question is from Dr. Hernan Vasquez Miranda. Are there any significant differences on a microbiome extracted directly from the gut versus an exposed and weather dropping?
This is an excellent question, and it's a nightmare because, of course, most studies are only getting fecal microbiomes. If they're lucky, they're getting fresh feces or they're getting an anal swab, and they have to assume that tells them about the community in the intestine, and in many species it's the upper intestine that's relevant, and so on. So there is actually an interesting area of research where you can kill animals to basically target the whole way down the intestine and also the feces and try and get a feel for what's going on there. In the foxes, we were fortunate. We got to go there at end of generation time, which means the foxes were basically being reduced in population size, and we could actually sample from the different things and see this. The real problem with post deposition of feces is it does change very, very fast. There's been a few papers that show, for example, they did something very similar. They went very simple. They got pig feces and they immediately extracted microbes from it, and then they put some in the fridge and some in the oven and some in the freezer, and they compared the community, right? And the communities were unrecognizable, like over these time series, right? Even when they'd just been in the freezer at -20 for a few days. And this is actually, I think, why there are these big synthesis papers writing about comparisons of microbiomes across many, many species. But if you factor in the sampling method variation, the data generation variation, the age of the samples, you actually lose a lot of the insight very, very quickly. To meet this, if anybody's interested, there is a guy called Anton Alberdi who works with me, who has got a project called the Earth Hologenome Initiative. And he is actually providing collaborators who work with animals special sampling kits. He sends them around the world where samples are taken in a very standardized way. Then back in Copenhagen, they're generating shotgun microbiome data from it. So it's a very, very standardized data set where actually they can then go back and revisit some of these big questions, but with perfect data. So if anyone's interested, Google Earth Hologenome Initiative, I think, or maybe Earth Holobarns Initiative, ehi.org.
We also had Hologenome Initiative.
Does that find something?
I'll put in the chat. Okay. Fantastic. I think that was our last question. So I thank you immensely for your time and for this interesting talk. And we hope we can get you here to Mexico anytime soon.
Thank you.
I'll have Dr. Susana Magallón back in the chat so we can close the section.
Yeah, well, thank you very much, Tom, for that fantastic talk. I was really wowed. It was very, very interesting. I am a botanist, and I really knew very little of the things you talked about, but it was extremely interesting. And I'm very happy to see that several people in the audience made very interesting questions. So I join Marcelo in thanking you immensely for the talk that you just gave us.
Well, thank you for having me. It was super nice of you.
Thank you very much.